Okay, just a quick look at the agenda today. We're going to kick off with a brief explainer video on precision medicine, just to give you a bit of insight into Life Sciences Hub Wales's role in how we're trying to develop the precision medicine sector. I am then going to do a brief intro to our challenge and the priorities that exist within it and how it will work. And then we have 3 fantastic talks lined up from key people from the precision medicine sector. We've got Sian Morgan from the All Wales Medical Genome Service, who's going to talk to us about the design of genomic revolution. We've got Tom Wilkinson from Swansea University, who's going to talk to us about host and bacterial biomarkers and sepsis. And Jonathan Thomas from the Medicines Discovery Catapult, who's going to talk to us about early diagnostics. And we will have then a brief time at the end of the agenda for some questions and answers, should any of you have them. Apologies, I've just realised that I've gotten to share with sound. 1 second. Hopefully,
Advancements in our understanding, detection and treatment of disease. Precision medicine holds the promise of customized diagnostic and treatment pathways, offering more personalized health and care. In Wales, our journey towards precision medicine is underway, with a strong commitment of intent from the Welsh Government. We're well placed to offer opportunities for companies to co-produce with the NHS and academia to develop integrated solutions for the global market. With unique strengths of genomics, imaging, intervention therapies, and point-of-care diagnostics, Wales offers opportunities for companies looking to collaborate. Benefits include access to data and sample storage, support for product development, internationally recognized academic excellence, close connections to policymakers, a stable population, and a devolved and integrated healthcare system. An agile and collaborative environment supports enabling parties to interact and work together to co-produce the technologies, diagnostics, and treatments for the future. Life Sciences Hub Wales is here to support and facilitate strategic partnerships. We're looking for companies who can bring innovative products or services that will help Wales achieve its ambition as a key influencer for person-centred care. Whether you're based in Wales, the UK, or internationally, let's start the conversation. Join us on our exciting journey in harnessing this potential, helping to make Wales the place of choice for health, care, and well-being innovation. Contact us at hello@lshubwales.com or lshubwales.com to learn more.
Fantastic. Hopefully everybody heard the sound correctly on that. Apologies for not showing the sound first up. So to briefly touch on the precision medicine priorities before we kick off our first talk. Obviously, with as you'll all be aware, being key people across the precision medicine landscape, that precision medicine is a huge area, and so to be able to address every single priority within that sector is, as I'm sure you'll appreciate, impossible. So what we wanted to do is to add some focus to that, and to do that, what we did is to bring key stakeholders from across the precision medicine landscape across Wales and actually further afield. Together to discuss what the key priorities within precision medicine were for the next 12 months. From that, what we actually came up with was a list of just over 10 key priorities for Wales. And then we then had a round of almost voting or prioritisation of those 10 key areas to come up with the top 4 priorities that we want to look at over the next 12 months. And that the precision medicine team here at Life Sciences Hub Wales, together with the partners across Wales, would like to look at. So those 4 priorities for you to see here, as you can see, are tools and technologies to support new or existing disease screening programs, tools or technologies that will enable big data analysis, particularly with clinical decisions in mind. Priority 3 is for tools and technologies that will support remote and or home-based testing of patients. And then the 4th priority for us over the next 12 months is opportunities for the repurposing of existing technologies for precision medicine applications. So essentially what will happen now is we have set up a challenge for which details these priorities on our Simply Do platform, which is already live, and that can be accessed by everybody. Further details for how you can see that challenge and get further information is going to be posted in several places. We're going to post it in a little bit later today into the chat function of this meeting. You will also receive information via email about how to join that challenge and how to get further information. Just a note, when you do log on to the Simply Do platform, you will have to register if you haven't done so previously, but as soon as you've done that, you will be able to see the challenge that is there. So I'm going to stop sharing and we're going to launch the first talk. Apologies, my computer is letting me down here. I think I've stopped sharing now. Fantastic. Okay. So it's with great pleasure. Apologies for those technical hitches. It is a great pleasure for me to introduce our first talker, and that is Sian Morgan from the All Wales Medical Genome Service. Sian, are you able to share your screen with us?
Can you confirm that you can see the presentation, Gareth?
I can, thank you, Sian. Yes, thank you very much. Please just remind everybody, if you do have any questions for Sian during her talk, please feel free to put them in the chat and then we can pick those up afterwards. Thank you, Sian.
Thank you, Gareth. I have had a warning of bad or poor networking. So can you please, if my sound does disappear, come back, can you sort of alert me if you lose me at any point? Okay, so my talk today is inspiring innovation in the Welsh NHS is the designer genomic revolution, another false promise. My name is Sian Morgan. I'm a consultant clinical scientist and the AWMGS laboratory director. This is my first IT hitch. It doesn't want to go down to the next slide. So I'll just end the show. And just do it again. Bear with me. Okay, apologies for that. So my first slide is from a publication that was shared earlier this year in the BMJ, and it was from a doctor called Dr. Morgan, Matt Morgan, who has sadly left the NHS Wales and has gone to work in Australia. And this is quite a harsh article where he describes the NHS as being a broken organisation, and he specifically discusses designer genetic drugs, the fact that we are offering false promises and cancers that will keep growing. So my talk today is about my experience working in the NHS Wales and my experience in delivering genomic testing. So this audience today absolutely understands why precision medicine is important. It increases survival rates, it targets tumours with greater accuracy, it mitigates unnecessary treatments and reduces prescription errors. But what does precision medicine mean for our patients in NHS Wales? But it means the right conversation being done at the right and asking for the right genomic test for the right treatment at the right time. And it's all about moving away from a one treatment fits all programme to a more personalised diagnostic programme. So what are the challenges for cancer diagnostic testing in the NHS? Well, you can have a group of patients all with the same tumour location, but they could all have personalised different treatment regimes based on identifying a biomarker in their specific solid tumour. But at the same time, you can have a group of patients all with differing solid tumours. However, they require the same treatment because they have the same driver mutation or variant in their solid tumour. And the challenges for us within the NHS is there's a lack of tissue when they take biopsies, and we need to give the genomic profiling reports back to the clinicians in a very timely manner. So an example of those patients who have differing solid tumour types, however, have the same variant or mutation driver, we call this histology independent. And the first drug that was published by NICE was for larotrectinib. This is and larotrectinib is specifically for biomarkers NPAP1, 2, and 3, and the incidence of this biomarker is seen across numerous tumours. But in addition to this, we need equitable access to genomic testing in a timely manner. We are all aware of the single cancer pathway, and that it's up to all of us to ensure that patients from the very first point where a cancer might be suspected receive cancer diagnostic tests and start their treatment within 62 days. And it's really important for us as well that all patients have access to this timely genomic testing as part of this diagnostic pathway across Wales. I'm going to give you an example of lung cancer and the evolution of systemic therapy for lung cancer. And these are the drugs that have been licensed in the last few years. And as you can see, the number of targeted therapies that are now being recommended and published are really gaining apace. And only just in 2020 alone, you can see the numerous amount of targeted therapies that were published for prescription. And aligned to all those different treatments, you need to identify the biomarker. And again, this is for lung. Approved therapies need biomarker detection for EGFR, KRAS, ALK, MET, BRAF, RET, ROS1, and NTRK. But we also need to be mindful that there are other emerging markers also coming down the track as well. So this is an ever-changing landscape that we have. And currently, the National Genomic Test Directory within the UK commissions all those biomarkers being tested as part of routine standard care. So current treatments available for our clinicians in Wales at this moment in time aligned to EGFR are these particular therapies for exon 20 insertions, for KRAS, for BRAF, for ALK, for ROS1, for RET, for NTRK, and for MET. It can feel like there's a massive wave of information and opportunities for our clinicians to absolutely align those treatments being very, very personalised to the patients that they are seeing. So what about testing and identifying these biomarkers? Well, there are a number of multiple tests that are available, from IHC to RT-PCR to FISH to NGS. But lots of different technologies will have differing limitations. And it is of no surprise, really, that ESMO issued their first recommendation on using NGS for advanced cancers in August 2020. So as you can see, you can have a one-stop consolidated test where you can test for all those rearrangements at the same time in the same tumour sample. And the reason for this is that you need the test. There are limitations around the different testing methodologies, but you have to be able to pick up single nucleotide variants, indels, copy number variants, fusion, splice variants, skipping, etc., etc. And that is why different technologies will have different limitations. So as you can see, there are now available on the market these big, large gene panels, and they're absolutely driving efficiency for the actual tissue, to reduce waste and to allow more patients to be identified, to get more eligible patients onto the right treatments. But to allow us to do that, we need to have the infrastructure to develop next generation sequencing. So where are we in Wales about setting up this infrastructure? Well, the Human Genome Project was launched in the 1990s, and the cost of the first genome cost $1 billion. And in 2001, we had the first draft of the human genome. And in 2007, the first people were being sequenced outside this particular project. And one of those people was Watson from Watson and Crick. And in 2007, the cost of his genome was £10 million. But you can see the cost per genome is outstripping Moore's law. And currently, within the NHS, we can now access a whole genome sequencing at a cost of £650. But we foresee those costs really being driven down over the next few years, because there are now more suppliers on the market, thankfully, that will absolutely drive that cost down in the short term. So we need to move away from single gene testing to absolutely testing more genes in a single test. And that is why it was really important for us to have the Genomics for Precision Medicine strategy launched in 2017, because we needed to build that infrastructure in the NHS healthcare system, because we could see there was a potential to save costs, improve quality of care by targeting treatment, maximising benefit and reducing side effects. And with that came an investment over 5 years. So we were one of those strategic partners to deliver on that strategy. So we are the Always Genomics Laboratory. We're a tertiary service hosted by Cardiff and Vale University Health Board, and we deliver genomics services for rare, cancer and pharmacogenetic testing for the population of Wales. We're regulated by UCAS ISO 15189 and we're commissioned by WISC. And currently in the laboratory, there are around 150 staff. And we're commissioned to deliver testing aligned to the National Genomic Test Directory for cancer. And that is to ensure equitable testing here in Wales compared to other patients across the UK. And as you can see, there are over 1,500 tests, cancer tests already on that test directory. The advent of and that infrastructure of building on NGS within NHS Wales has absolutely revolutionised what we now deliver across all the services we have within the laboratory. But today I am just going to continue to concentrate on precision medicine. So at the time, even back in 2019, we had a few different technologies running in the laboratory, including a small NGS panel, RT-PCR, pyrosequencing, FISH, etc. And we were realizing the need to go into quite a large multi-gene solid tumour panel was required for our services. So we went out to tender and we launched the Cuscodi service in 2021. So the Cuscodi service is a company service for genomic oncology diagnosis. And this is a picture of one of our patients who went on the camera for this piece for the BBC, where she described her journey, where she had a precision medicine and an outcome, and she'd had this precision medicine quite a few years, so her outcome was really good, and she talked about her own experiences. So it's absolutely critical that we drive economies and efficiencies. So we validated the 500 gene panel, and our validation process is accredited to a framework for the validation and verification of clinical molecular genetic testing in the UK. And we have a single workflow now where we add our multi-tumour samples on the same workflow, and we're just about to add our hematological samples to this workflow as well. It doesn't matter what the solid tumour is, it goes onto the same workflow. We extract both DNA and RNA, and we sequence 500 genes across all samples that we receive. But the bioinformatic analysis will only allow us, the pipelines will only allow us to currently report against those targetable mutations where we know that there's a target treatment available. For example, for lung, we currently report EGFR, BRAF, KRAS, EGFR, ALK-MET, RET-ROS, NTRK 1, 2 and 3. And we upload our results onto the Welsh Clinical Portal. So this is up-to-date data. We've started the Scotty service back in August 2021, so we haven't run this service yet for a year. We currently run lung, colorectal, melanoma, GIST, thyroids and glioma on this particular single workflow. We've got a lot of work to do because there are other solid tumours we need to add to this workflow. We've run 1,500 DNA samples and 465 RNA samples have undergone this testing. NGS analysis has been successful in 99% of DNA and 87% of RNA samples tested, and a variant has been identified for available targeted treatment in 37% of patients, including 7 RNA fusions. So this particular strategy around economies and efficiencies will allow us to be able to react efficiently to any recommendations that might come from, for example, NICE around any new drug that might come for our clinicians to prescribe. So it's a strategy that it's a one-stop test. You get all the testing done simultaneously at the same time, and it's a strategy that we see that will allow us within the NHS Wales to have a really good pipeline up and running for the short, medium and hopefully into the long term. We can't do this without the support of the All Wales Genomics Oncology Group. The AWGOG sits under the Wales Cancer Network. We established this group in May 2020, and it aims to ensure that there is a multidisciplinary, coordinated national approach with defined clinical input to the development and introduction of new oncology-specific genetic services within Wales. So I've touched on something very specific in the talk today. We've talked about the 500 gene panel for our solid tumour diagnostic services. There is so much more that we're doing in service that obviously I can't cover in a 10-minute presentation today. But we're all aware of the work that's happening within AWMGS around ctDNA and the work that has started around whole genome sequencing as well. There's a lot to do, and as I say, it's quite targeted what I have fed back to this group today. So what's next? We need to continue with a whole UK genome approach, and you will all be aware that the genomics delivery plan for Wales is currently out for consultation. The Genome UK was published in April 2020. We've got the NHS 10-year plan. We've got the quality statement for cancer. Timing is absolutely everything for our cancer patients in Wales, and we will need to continue to try and follow and be able to ensure that we have got testing in Wales aligned to these policies and UK direction. As I say, we've got a lot to do. Thank you, Diolch, for listening today, and I'm really happy to take any questions from this presentation.
Fantastic, thank you very much, Sian. Yeah, fantastic talk, and as Chris has highlighted in the chat, a great, a hugely, an amazing resource for Wales, and fantastic work's going on. Does anybody have we have time for a quick question for Sian? Does anybody have a question for Sian? Either happy for you either to put it in the chat or just raise your hand so we can see you. I think I saw a hand go up there. Yes, go on.
Hi, my name is Tan Rasab. I'm the CEO and founder of a company called Sigenics. I'm the chair of the Genomics Committee at the Wyss Institute at Harvard, and our plan is to how do we make targeted panel testing much more accessible and eventually in the clinic instead of centralized labs. And I started my company 2 years ago, and I advocate a lot of this on LinkedIn. And the people who are most advocate supporters of mine in the UK are from Wales. So we're hoping that when our first prototype device comes available, that Wales will be the first region or first place in the Western world will be our place where we actually do our trials. I mean, we're going to do pre-trials at probably Boston Mass General Hospital and the US Army, but in terms of commercial deployment where we can help Sian Morgan and her colleagues to maybe split some of the testing to be done in the clinic rather than centralized lab. The advantage with what we're doing is it will significantly reduce the time to results to about 26 hours for most of the panels that Sian just discussed. So I'm really, everything she said and everything you guys are saying it's music to my ears. So thank you for inviting me to this event, and thanks for Sian for presenting what she presented today.
Thank you, Tan, and really exciting to hear more, and really happy to meet up outside the meeting today to see what opportunities there are.
Thank you so much. Thank you, Sian. Will do.
Yeah, thank you very much for that question, Tan. Yes, of course, happy to be involved in those discussions, and looking forward to supporting where possible. We've got one more quick question before we move on in the chat, Sian. It's from Chantel Patel. Have you seen it?
Yeah, absolutely. A great question. Obviously, I've just concentrated on cancer today. So obviously, there's a lot of treatments coming out as well for rare disease. And where we are with rare disease at the moment is obviously we are doing whole genome sequencing, from different technologies, but we are doing whole genome sequencing as well for rare disease as part of the service here in Wales. But we will all be aware of the big national project that will be starting in England, probably either the end of this year or next year. And I'm keeping everything crossed that we will be part of that huge national programme, and that's the newborn screening programme, where there are obviously precision treatments that may be required quite early on in life. So there is plenty going on in the rare disease forum as well. Hopefully that gives a quick answer to Chantal, but thank you for a great question.
Fantastic. Thanks for that, Sian, and thank you for the questions. We're going to move on, but if you do have any more questions for Sian, please put them in the chat, and we can forward those on and address them after today. So next up in our talks is Tom Wilkinson from Swansea University, who is going to talk to us about biomarkers to predict sepsis. Thank you, Tom. I will hand over to you.
Hi, Gareth. Can you hear everything?
We can hear you, and I can see your slide.
And you can see a slide. That's wonderful. Okay. So I'm Tom Wilkinson. I'm Associate Professor in Microbiology and Infectious Disease at Swansea University. Bit of a change of tack now. I'm going to go into infectious disease, and I want to tell you a little bit about some of the strategies we've used to try and develop new biomarkers to predict bacteremia and associated sepsis. So pretty much sepsis is one of the leading causes of death worldwide. There was a great study in 2017, pretty much suggesting that there are 49 million cases throughout the world, leading to about 11 million deaths. So when you think about it, that's about 20% of all annual world deaths. Problem with sepsis, bacteremia, is that it's a very complicated syndrome because it has dysfunctions in both inflammatory pathways, anti-inflammatory pathways, complement and coagulation pathways, and all of those are all mixed up together. There's a dramatic correlation between the time to antibiotic administration and death. And that means the window of opportunity for therapy is incredibly short. Now, the current biomarkers that we have, things like IL-6, CRP, procalcitonin, are very, very inconsistent, and there's a lack of discriminatory power to be able to predict which patients will go to the severe form. So, of course, early prediction then and some sort of risk stratification is really urgently needed. And I want to share some data today on some strategies that we've used to be able to try and address elements of this. So important organisms involved in bacteremia and sepsis include Staph epidermidis, Staph aureus and E. coli. But I want to focus a little bit more on E. coli bacteremia because that's a problem in Wales. In particular, if you look at the graph here, it's a little bit old now, maybe 2018 numbers, but the Hywel Dda Health Board is probably had one of the highest incidences of bacteremia. It's a relatively large geographical area, and probably the latest figures are around more the 80-85 per 100,000. But one of the reasons for this is it was a very good surveillance. It has a very good surveillance program. You can see the reference there from Mike Simmons. So we thought this was a good opportunity to be able to collaborate with the NHS, and maybe to be able to identify new potential biomarkers. Now, I don't know the audience, but just a little bit on E. coli. I won't go too microbiological, but just to say E. coli obviously comes in at least 3 flavors, if not many more. But E. coli can be benign and pathogenic. And if you focus on the schematic here, 2 big groups of pathogenic E. coli include pathogenic E. coli that stay in the gut, but obviously pathogenic E. coli then that go away from the gut, or extra pathogenic E. coli. Now all of these carry virulence factors that aid them to extra intestinal spread, but often it's not easy to predict which E. coli will lead to a particular systemic infection. Okay, so the strategy we used on a relatively small scale, but at the scale at how well our health board is, we started a calibration going, and we isolated, or we took 157 blood culture positive E. coli, and we did two things to them. We had one pathway, if you look on the right, where we started investigating phenotypes associated with that E. coli. And the phenotypes we started looking at were, for instance, their ability to produce an inflammatory response in whole blood, their ability to grow in serum, but also we went back to patient records and started looking at patient data, so we get maybe better clinical definitions of where these E. coli came from. Then the second pathway that we had there in green is that we started sequencing these and doing some basic genomics so that we could identify the virulence factors and antimicrobial genes. And so then what happened is we put the green and the blue together so that we could start focusing on gene associations, with the hope that we could actually push on and look and develop maybe some novel biomarkers within that collection that we could push maybe forward for more clinical validation in patient serum samples and beyond. So something very short on gene association pipelines. Everybody does this in slightly different ways. However, the way that we did it, we organized 157 E. coli isolates into groups. So that might be on the source of infection. So for instance, maybe a urinary tract source versus a gut source. It could have been maybe ex vivo data, so from our whole blood model. But it also could be just serum resistance, the ability of these isolates to grow. And then we compared the genes that were present and absent in each of these groups, and we totaled them up. Now, as I said, numerous methods and arguments can be had over how you do this. This is a very short explanation of how we did this. We assembled the genomes, we annotated the genomes. In other words, we looked to see how many genes each genome had. And then we created a huge list of all the genes we had in E. coli. And we started assigning how many each of the groups had of each particular virulence factor. So what I'll do is I'll present a little bit of data on some of the phenotypes, and then obviously go on and look at some of the links that we started to make with general virulence factors or total virulence factors, but some specific ones as well. Right, so this is the patient cohort that we had. So I don't think there's anything dramatically different in this patient cohort. There's some biases in some places because it's a relatively small sample. But the average age was about 75, is probably what you would expect. Obviously, big age range there because we've got a baby in there as well. The gender was pretty even. If you look at the table on the left here, gender was pretty even. The sources of infection, there was a tendency to a bias, obviously, in urinary tract infection and developing urosepsis. If you keep on going down the table, about half of the patients ultimately went from a bacteremia to sepsis. Again, about half were nosocomial infections over community-based infections. And we also found some comorbidities associated, which were cancer, diabetes, and urinary tract infection. And the mortality rate in our cohort was about 14%. I won't go through all the AMR properties, but maybe just to highlight a couple. If you look at ampicillin and amoxicillin at the top there, we get about 50% resistance. And if you maybe go down to pip/taz at the bottom, piperacin and tazobactam, about 10%. So the first and second line antibiotics tend to give, this is what we probably expect in general. And then if we go on and look at multi-drug resistant, the incidence was probably about 10 to 25%, depending on your definitions there. So we went on to look at how this collection was able to induce early inflammatory responses, so trying to mimic that early inflammatory response in bacteremia and sepsis in a whole blood model. And we found differences when we compared the bacteremia isolates with some of our non-pathogenic isolates. So for instance, in particular in graph 2, you see IL-8 significantly higher and MIP3, a chemokine, an early induced chemokine, also high. Interestingly enough, no changes in IL-6. IL-6 is a biomarker currently in sepsis. And that's probably associated with the fact that this is just whole blood, and much of the IL-6 response is obviously associated with the liver and the acute phase response. But here we've been able to identify some groups that we can use in our GWAS and association pipelines later on down the line. The other thing that we went on to do is we also looked at phenotypes in serum with these isolates. And essentially what we've got here are 3 graphs. If you look at the dark curves first on each of these graphs, you can see in the left-hand graph, I won't do colours just in case. If you look at the left-hand graph, you can see these are resistant isolates, so these grow very quickly from 2 hours. The intermediate isolates in the middle, these grow a little bit later from about 8 hours onwards. And the sensitive isolates are killed straight away, and you don't see any growth in these. This was a complement-dependent effect, so this killing was related to complement. If you heat treat all of those samples, many of them tend to be able to grow then without the complement in place. So again, this was 3 groups, another 3 groups that we could add into an analysis later on down the line. And this is, you remember, using all of the isolates that we've got, not just 1 or 2 strains like you might see in many microbiology experiments. We also went to try to look at any correlations or links between the cytokines in whole blood and some of the patient mortality data that we identified. But we really couldn't find any differences in any of the cytokines that we identified there. What we did find, however, was there was a very wide distribution in some of these responses. And that's probably something that we'd like to look at there, that, you can maybe start to stratify responses based on high, medium and low cytokine levels for the future, and then look at the genomes associated with those bacteria based on those. So that's something for the future, but we could find no difference in the isolates that caused ultimate death or not. We also went on to look at just the total virulence factor expression in these isolates. So the black bar on the graph there are bacteria, our bacteria isolates, and you can see that these have significantly more E. coli related virulence factors that are pretty much established, significantly more than water isolates, probably no surprise there. But if we go to the 3rd bar, the green bar there, there's very little difference, in fact no difference, between bacteremia isolates and asymptomatic bacteriuria isolates that we found, with respect at least to the total number of virulence factors. And that's really underlying how difficult the E. coli situation is, and how getting more specific identification of really invasive strains over strains that really do not ultimately cause disease is important. So if we got more specific, what we've done here is we've gone on, this is part of a huge table. There's a lot in this table. What I really wanted to focus on is obviously the first column and the 3rd column. The first column is individual genes, and the 3rd column is the association. So the association here is where we're looking at the phenotypes that I've talked about in some of those graphs. And here we're starting to look at how strong that association is. And so what we've got is we've got phenotypes here, for instance, associated with the amount of cytokine produced in our models, with mortality from the clinical data, nosocomial versus community infections, or indeed their origin of infections such as abdominal or urinary associated. So in other words, individual genes can ultimately be associated with a group or phenotype, and you can obviously allocate a sensitivity and a specificity to those. But this allows us to round down huge numbers of genes from 10,000 genes to probably about 50, 100, maybe a little bit more. And these then can be used as panels or investigated further at the basic science levels. So some of these genes deserve further investigation, essentially. So I think what we've done is we've we developed a new collection of E. coli from Hywel Health Board, and we investigate traits and phenotypes in those strains from all sorts of backgrounds. They could be from ex vivo models, they could be from in vitro models, or they could be from patient data. And essentially, once we've got those models and groups, we can start to further validate those targets in the next step, in particular, probably using patient samples and real sera then from patients with bacteremia or indeed sepsis. So importance, I think, of these findings is that I think we're using some clinically relevant strains of E. coli. I think there's the potential here for cost-effective and practical targets that I think could be used by the NHS, in particular when we talk about repurposing some technologies. Some of the targets here are perfect for new panels of maybe PCR for virulence factors. So there's a possibility then for us to push towards improving risk stratification. And I think this also probably adds to a little bit of basic science evidence on looking at E. coli host pathogen interaction. So I probably just about up to time. So I just wanted a few acknowledgements of this work. This is this work that's funded by the Welsh Government, Healthcare Research Wales, and we thank those. Part of Matt Lewis's PhD. A big thank you to clinical colleagues in Hywel Health Board, Mike and Jane. But please email me. We're a gang that love infectious disease. We want to find solutions to infectious disease, and in particular things like bacteremia and sepsis, where there's a quick decrease in patients very early. So I'll leave it there, Gareth, and I will answer any questions.
Fantastic. Thanks for a great talk. Thank you very much, Tom. We actually have one question already in the chat, so I'm going to kick off with that one whilst you, if you can stop sharing your screen. We're all having tech issues today, aren't we? But that's fine. So we've got a question from Matthew Thomas, who is asking if there is a role for AI in candidate biomarker identification.
There was a role for AI in something else. Sorry, Gareth, I missed that.
That's all right. Is there a role for AI in candidate biomarker identification?
Yes, without doubt. I think we should have that discussion. I think without doubt, please email me and let's have the coffee and that discussion. I think certainly that's the case.
I've got another question. Another question from Matthew, actually. Matthew, do you want to come on quickly and just ask this question? Is that easier, or?
I'll happily do that. Yeah, sorry, it was badly worded because I edited it after. Yeah. Anyway, essentially, we saw that. I think what you were describing is the interaction between the host and the pathogen genomics. Or did I miss that?
I'm sorry. I'm literally. I heard pathogen genomics. I'm really sorry. Yeah.
So if I'm just checking that I heard correctly, what we were looking at there was a description of how an interaction between host, as in the human genomics, and the pathogen genomics, yes, gives you a sense of, you know, what. Well, it gives you a whole lot more intelligence. Is that right?
Yeah, so I think so. The genomics that I presented there was just associated with the pathogen. Okay. The host work was all phenotype. There was no. We didn't have the ethics to go further into getting patient tissues there. It was all medical records. Okay. So there's no genetic. There's no genetic data from the patients there. They're all anonymized. That might be the next step. It was all genomic data from bacteria. Yeah. Yeah. Great. Thank you. No, that's probably because I'm fairly new in terms of understanding genomics and you know the meaning of terms like phenotype, etc. But that you've clarified it for me. Thank you. I was just thinking, you know, COVID-19, big thing, right? And you know, it's been in the news for everybody about how, you know, certain groups of the population were more susceptible to the harms of COVID-19. Others brushed it off more easily. I just wonder, you know,
could the interaction between the host genome and the pathogen genome be studied retrospectively now, if it hasn't already been done? And can we learn from that a general approach to all pathogens, and an interaction between pathogen and human genomics, so that we could be better armed to deal with the treatment and so on?
I mean, I think some sort of certainly screening in these early stages of infection. So screening, for instance, for things like our key receptors that are involved in sensing organisms. So, for instance, pattern recognition receptors like toll receptors. Certainly screening for these, and people are doing this. These are key receptors that are involved in identifying E. coli or identifying Staph aureus. And whether these are different and whether their binding is different, for instance, between patients, and it's known that they are in some cases. So this sort of screening, I think, is really important. Yes, I think that's certainly good potential. Yeah.
Can I just interfere? Rather, I was just actually reading a paper today that they've identified 1,000 genes that most humans carry that actually have a direct impact on how COVID affects you. So it depends on your ethnicity, your immune response, and so on. So what Matthew has raised is actually a very good subject to investigate further. And I think eventually we will have personalized, antiviral drugs as well as antibiotics. So what you might be prescribed, Matthew, may be different to what I have, because my background coming from the Indian subcontinent makes me more risky, makes me a higher risk for certain pathogens against others, where you will probably have a higher risk against other pathogens. So it is a really important discussion, I think, which is being done in many research labs around the world.
Right. So the importance really then of sequencing, the population at scale, so that we can, have that information available in order to do the analysis between and across all of the different possible harms that can come to us, so that we can work out treatment. Doing it at scale and getting the economies of scale means we've got to have big data, right?
Yes.
Yeah.
And sorry, go ahead, Thomas. Sorry, I don't mean to be overtaking.
No, I was only going to say you might talk about sequencing your whole genomes in human, but I sometimes think you don't need to do that. I think maybe just sequencing the key parts of maybe the immune system that is sensing some of these, that are involved in some of those. You mentioned the 1,000 genes. Well, maybe it's those genes that you sequence, rather than thinking of being cost effective. But it might be more cost effective to do it all anyway, thinking about it. But that's all I was going to say. Sorry.
There is actually a paper published by Johns Hopkins and the World Economic Forum, which came out last year, and they recommended that panel testing is the most effective way of introducing genomics into the clinic. Population genomics, you do these big, huge centers where, they will do longitudinal studies, which will take 10 years and so on. But if you want to do fast testing and fast, precision medicine implementation, do panel testing, because it's much more effective,
Panels the way forward,
and it's faster.
Fantastic. Thanks very much. A fascinating conversation, and I'm loath to have to to close off the questions, but keen that we pick up this conversation afterwards between all of us and continue it. But thank you again very much again, Tom. Time is running away with us, so I'm going to move on to our third talk for the day. Jonathan Thomas has joined us from Medicine Discovery Catapult, and he's going to talk about early diagnostics. Jonathan, are you able to share your screen? Fantastic, I can see your slide. It's a good start. I just need you to unmute for me.
There we go, that old classic. Thank you very much for letting me take part today. Can I just make sure you can see the presentation correctly?
It's switching between screens at the moment. I can see it for a second, and then it's switching. Now I can see it.
I was having a bit of difficulty. How is that?
Yes, I can see your slide now. Let's hope it sticks.
Yeah, as you said, we're all having some technical issues here, I think, aren't we? So that should be the screen. Excellent. No, I mean, it's always a bit of a challenge having the sort of the last presentation of a series, I guess. Even more so when you're following two very technically astute and eminent colleagues, and again, even more so when some of what you wish to talk about has apparently been covered. So I'm probably in more technical detail. So I put a presentation together here, and you know, I should probably highlight, you know, this is very much from my personal perspective and experience, having spent a number of years within the UK diagnostics sector before joining the medicines discovery catapult. Now, I must confess, I do like to take a bit of a simple approach to things. So, in terms of, you know, what we generally mean by precision medicine, you know, as you may already know, or picked up from the previous presentations, you know, it's essentially using the individual's data, you know, that might be sort of clinical, based on molecular biology, lifestyle information, etc., to really sort of guide disease treatment and prevention. So, as Sian mentioned, you know, it's very much a move away from that one size fits all approach to one of more sort of targeted therapeutics. But the one thing it's not is that sort of personalized medicine approach where you're actually creating drugs that are unique to a particular patient, but it's using the information that can be generated, you know, to really classify individuals into different subpopulations, you know, which that may be based on susceptibility, you know, to a particular disease, you know, its presentation, prognosis, and it also be used how people predict how they may respond to a particular treatment. As I mentioned, I spent a number of years within the UK diagnostic sector. The last, well, predominantly within the lateral flow world. As you can imagine, the last 2 years have been a bit of a roller coaster. But early diagnostics, as the name suggests, essentially, you know, it's a way to detect disease much earlier than current practices may allow, and having that information more readily would allow more targeted therapeutic interventions, and, you know, really does shift that clinical approach from a more reactive position to prevention, you know, by using data to classify people into specific subpopulations, look at who's more prone to what disease, and what sort of disease prevention strategies could be activated within certain communities. You know, and, you know, as you can imagine, the early diagnosis, early detection enables the correct therapies to be used at an earlier time as possible. Knowing the data around the subpopulation, you know, will allow you to avoid any therapeutics. There may be a sort of predictable adverse side effects, and thinking from a sort of economic perspective, by eliminating that trial and error model, you know, that comes to therapeutics, you know, that can reduce healthcare costs, but more importantly, really, you know, move away from that model, which can undermine patient care and undermine confidence, you know, in the treatment that a particular person may be receiving. Biomarkers are something very close to my heart. It was a great presentation from Tom, and it's one of the platforms within the MDC that I work closely with my colleagues. Now, a simple definition of you know, a biomarker, you know, it maybe described as an indicator present in the body or excreted from the body as a response to a disease. Obviously, a biomarker can be used to detect and diagnose a disease, and in many cases, whilst the individual is asymptomatic. Now, modern biomarker discovery very much does use a sort of multi-omic approach. For example, at MDC we have a variety of equipment from NanoString, including the Genomics, to support the drug discovery sector within the UK. But that opens up proteomics, looking at transcriptomics. I mean, it really is a bit of a game changer in terms of the data that can be generated and how that can be used. So, in summary, before I go on to some sort of real life cases, I mean, and examples, I mean, in terms of early diagnostics, the aim is obviously to diagnose the disease as soon as possible, do it in the most sensitive, the most specific manner, provide information, that it's going to give relevant input with regard to potential prognosis of the disease, and really feed into to the selection when it comes to whatever therapeutic drugs will be chosen. Now, in terms of the types of early diagnostics, I mean, they are currently a mix of very different sort of platforms, and they will continue to be so. You have the likes of clinical laboratory-based equipment, the may be closed for specific reagent kits, or more open to look to use a variety of different assays. Maybe looking at sort of imaging techniques. So there really is a variety of potential technologies. But the potential to really sort of center that delivery of care around the patient is thought it will lead to an expansion of point of care testing, and potentially, as with some examples and some types of assay, moving into more sort of home testing in environments, really sort of decentralizing healthcare. Now, I should apologize to anyone from North Wales, and I assure you, even though I have the accent I have, I'm not completely South Wales focused, but these are three companies that I've had some connection with over the years, and they're just three examples of companies based in South Wales involved within the diagnostic industry, and to just give you a bit of a flavour really as to what's going on outside the academic labs, I guess. So, I mean, for example, EKF is a Cardiff-based company, and has supplied a huge number of analyzers to more than, I think, over 100 countries. Now, the one thing that they've noticed, though, is the uptake actually within the UK is not as great as other countries, because there's a bit of a challenge to get these systems into the NHS and to get them to those sort of satellite NHS establishments and locations. So even within the industry, assays can be developed, would provide diagnosis at a much earlier stage, that would allow someone to manage their own well-being, but there may be that reluctance, and there's not really that sort of pull from the healthcare industry, healthcare sector, sorry, to actually implement those changes. So in some countries there can be a bit of work needed to really push those things forward. My Health Checked is another Cardiff company, and they are really more in the realm of molecular biology based tests that may be done at their laboratory, at another laboratory, but in terms of the sampling kits, that's all done at home. It may be a sort of cheek swab, saliva sample, etc. But they are very much looking to expand their offering to put the sampling and ultimately the testing into the hands of their end users, and by combining that with sort of healthcare app of some kind, really sort of decentralizing healthcare and sort of well care, as I think they describe it. As I said, my background previously has very much been in lateral flow, so BBI's company that was again was Cardiff based, but is now more now located up in Kremlin, and this is the company that is involved in the contract development of lateral flow assays. And again, from speaking with the group product manager, David Fraser, I mean, he very much believes the experience of the last 2 years and will really engage the general public to adopt more home care testing. There's obviously the distinction between the application, and the application for an LFA has to be right. Is it currently more suited as a rapid screen as opposed to a strict diagnostic tool, because perhaps of the lower level of detection and the specificity of the assay. Now, there are innovations coming through, and we're seeing more and more PCR based assays really being packaged into something which is getting more and more like classical lateral flow assay. So it's certainly thought again that this will be an area where we will see perhaps not an explosion, but definitely a significant increase in the types of testing that can be done at home, and giving people that sort of personal management of their health. So I know we're over the 3 o'clock, but again, if anyone has any questions, please feel free to ask.
Fantastic. Thank you very much, Jonathan. Great talk. And yeah, we do have time for one. I appreciate we're overrunning, but if anybody has one, I can allow one question for Jonathan. If anybody has one, if you want to put your hand up or quickly throw it in the chat. I can't see any hands going up or anything. Okay, I think you've got off lightly there, Jonathan. But what I will say to everybody, if anybody has a question afterwards, please forward it to us and we'll make sure it gets passed on. Thank you very much again, Jonathan, for a great talk. I appreciate I've held you beyond our allotted time, so I'm going to very, very quickly wrap up. I'm going to try and share the right window now. We don't know what's going to happen with these, but here we go. Hopefully it's just for me really to say thank you to all of our speakers, to all of you for attending. It's been a great presentations from everybody. I can see we've got lots of conversations that we need to pick up after this as well to continue those conversations. I'm very keen for anybody who's interested in discussing any of the topics that we've covered today further. Please reach out. We're happy to pick up those. Hopefully, magically, someone is going to paste the link to the challenge, the Precision Medicine Challenge, into the chat momentarily. There it is. Fantastic. If anybody would like to submit solutions to this challenge, of course, I encourage it 100%. Please have a look. Please submit any solutions you have to those 4 priority areas of the challenge. And if anybody wants to discuss either those priorities or other priorities which you have in your businesses, please get in touch with us. Happy to discuss further. And thank you all for attending, and I hope you enjoy the rest of your afternoon. Thank you very much, everybody.